Everyone, thank you for coming today for our special Thursday seminar of our department. We have Dr. Rich Mizell. Is that right? And I'm very excited to host him, and we had a great day talking and meeting with lots of you and students as well. So Rich began his scientific career at Cornell University, where he did his biology undergraduate degree. He then moved on to a PhD in genetics at Pennsylvania State University. And that was followed by a postdoc back at Cornell with Andy Clark in the Department of Molecular Biology and Genetics, where he developed his interest in sex chromosomes and chromosome evolution. Since starting his lab in 2013 at the University of Houston, where he is currently an assistant professor in the Department of Biology and Biochemistry, Rich has done really interesting work on sex determination, genetic variation, and sexual conflict. His work is funded by a career grant from the NSF that started just in 2019. And today he'll tell us about his work where he's looking into how sex chromosome differences or other genomic and environmental contexts influence gene expression.
In terms of thinking about what constraints can be put on context-dependent gene expression, and I'll get to sort of what I mean about that. Oops, that's not it. The first thing I want you to think about is the fact that organisms are not these static creatures, right? They change over development. So from egg to adult, for example, in an animal, you have a huge morphological, behavioral, physiological restructuring. It leads to tissue differentiation. You get these different organs, organ systems, structures that have to happen through that developmental process. We see differences between males and females as well in terms of behavior, physiology, anatomy, all that good stuff. And these are sort of programmed developmental processes that give rise to this, but we also have sort of environment-dependent changes that occur. For example, when you're exposed to a pathogen, you need to elicit some immune response to combat that pathogen, or else you can get sick and even die. Same thing with abiotic stressors. So one thing we think about a lot in the lab is temperature as an abiotic stressor. And so climate, temperature can be hot, can be cold. You guys are familiar with cold. I'm in Houston, I'm very familiar with hot. And so all of these things are acting on an organism, causing it, the phenotype of the organism to be plastic or to have programmed changes. It's all going on under certain constraints. And the most obvious constraint is that the organism has to accomplish sort of anatomical, behavioral, physiological diversity across itself, across development, across sexes, across different environments, using a single genome, right? So you have one essential blueprint that you have to differentially deploy in different contexts. And that can create a constraint because you have to regulate that differential deployment. And so kind of taking us all back to maybe for some of us a long time ago, think about how does this work? Of course, gene expression changes are an important component, something a lot of us in this room think a lot about, but just to make sure we're all on the same page, right? You have a gene, it's transcribed, that transcript can be made into a protein, and that differential deployment of that gene in terms of how it's expressed or how it's translated can give us that diversity of phenotypes across development, across abiotic and biotic contexts. And to regulate that, there's sort of built-in cis-regulatory sequences that are always present adjacent to the gene, and then there's things that can be added or subtracted in different cells in order to change expression. So things like transcription factors can be present or absent, and can modify the expression of genes. You can also have histone modifications that can promote or inhibit transcription, translational regulators as well. And so we'll just call all these things trans factors. And these are the things that differ, for example, between males and females, or that change across cell types during development, or that can be present or absent under different abiotic or biotic environmental conditions. So these trans factors then are which you can sort of change to differentially deploy the same genome across different contexts. And so that's, that's sort of well understood. What I'm telling you, for a lot of you, is like, duh, that's obvious. And we can see this in, if you look at gene expression, say between a male and a female Drosophila. And so one of the first experiments that did this, nearly 20 years ago now, did this with microarrays, and we said, okay, what genes are upregulated in males, and that's the blue chunk here. What genes are upregulated in females, that's the red chunk. And so these are genes that are being differentially deployed in males and females in order to produce the different phenotypes in the two sexes. And so, this is, okay, that's how you deal with it, right? There's a constraint, you have one genome, you deal with it by differentially regulating the genes under different contexts, and then you can differentially deploy the genome. But I don't think it's that simple, because a lot of the ways that these things are differentially deployed is shared across very unrelated contexts. And so here's a very old illustration. We know a lot more about how this works now, but the general theme is the same. This is how antimicrobial peptides are regulated in Drosophila. And so everything on the bottom here, Drosomycin, Cecropin, et cetera, those are all antimicrobials. And there's, essentially here it's illustrating, there's two main regulatory cascades. There's the toll pathway on the left and IMD on the right. And if you're a developmental biologist, you might say, no, the toll pathway is the signaling pathway for embryonic development. And so what we're interested in, and something we really haven't dug too deep into yet, is to what extent does this shared pathway in two very different contexts constrain the organism's ability to specifically respond to those very different contexts? And basically the question is there a crosstalk across these pathways such that you get sort of a noisy response when you get a particular stimulus? And if you look even closer at the toll pathway, which is here, there are regulators of the toll pathway like this serpin, SPN27A. It's involved in melanization, and it also regulates input into the toll pathway. So this common theme of basically there's a limited number of signaling pathways, there's a limited number of pathways in which you can communicate information within cells, yet the cells need to be able to respond to all sorts of different contexts, use those limited pathways, and produce specific responses to those contexts. We're interested in understanding, to what extent does that constrain the ability of the organism to have specific responses. And I'm actually that's all I'm gonna say about this. I don't have any data here. I just wanted to sort of plant that seed in your mind. And it's something we're interested in looking to more in the future. What I do want to tell you a little bit more about in detail is a constraint of how many copies of the genome that you have in the cell, right? So for most genes in most cells, there are two copies of the gene present, right? For a diploid organism, okay? So you imagine you have two copies of the gene, and you can express from both of those copies. Of course, some things are imprinted, and they can only express from one copy. But for many genes, right, you have two copies. Now, we know that that creates a constraint, because we see that in some cell types, the organism goes out of its way to replicate the genes involved in certain functions beyond two copies per cell. And so if anyone here has ever looked at a Drosophila salivary gland dissection and looked at the polytene chromosomes in there, those are many, many copies of chromosomes stacked on top of each other. And one thinking is there are certain genes that need to be produced at very high levels in the salivary gland, and that's why those chromosomes are what we call endoreplicated within cells. Another example is in the Drosophila ovary, the chorion proteins, which are important to the egg coat in the ovary, are endoreplicated within cells many times, because the cell basically needs to be, certain cells in the Drosophila ovary need to be chorion protein producing factories. And so they need to make all of these chorion proteins in order to do that, they need to make many copies of the genes in the cell. So it's clear that there are certain cases where having only 2 copies of a gene in a cell creates a constraint, and there are certain cases where the organisms have come up with workarounds in those specific cell types in order to deal with that constraint. And so we wanted to approach this systematically, and so we decided to say, okay, well, one situation where this is likely to occur is on the X chromosome in males, where the entire X chromosome is haploid in males, if you have an XY system where the X and the Y are differentiated, and the X chromosome is diploid in females. And so can we find patterns on the X chromosome that indicate that that haploid dosage of the X in males is creating a constraint that is preventing, say, highly expressed genes from being on the X? And so the first place we looked at this, or we had looked at this, was in reproductive tissues. So we decided to contrast male and female reproductive tissues in Drosophila. So this is the male and female Drosophila abdomen that I'm showing you up here. And what we did is we took available data that had been collected, so this is actually microarray data, from many Drosophila tissues, and we asked what genes are specifically expressed in, say, ovary, or specifically expressed in spermatheca, 2 female reproductive tissues or organs. And so the spermatheca is a sperm storage organ. The ovary is the organ that produces all the eggs in the female. And what we saw in the ovary is that there was a significant excess of genes on the X chromosome that are specifically expressed in the ovary. So the way you read this plot is the dashed line is the percent of genes we would expect to find on the X chromosome if genes were randomly distributed across the genome, and the bar indicates the actual percentage of genes on the chromosome. So ovary, genes that are expressed specifically in the ovary overrepresented on the X chromosome. And there's all sorts of explanations for this that I'm not going to get into. What I want to highlight is the situation that's going on in males. And so we looked at testes, which produces sperm, an accessory gland, which is sort of like the Drosophila prostate. It's a protein-producing factory that makes seminal proteins that get incorporated into the ejaculate, and they're essential for stimulating ovulation in the female. They're also important for preventing the female from remating. So the males are under selection to produce a lot of these things, transfer them to females, and realize a fitness benefit because the female will have offspring, and they will be that male's offspring. And when we looked at the accessory gland, we saw there's a significant deficiency of genes specifically expressed in the accessory gland on the X chromosome. And this is consistent with, this is annotation blind, right? We don't care, we didn't say what these genes were doing. We just said, are they specifically expressed in the accessory gland? Previously, people had shown that genes that had been annotated as accessory gland proteins were also deficient on the X, okay? So we thought, well, maybe then there's a dosage limit. And you don't want to be on the X chromosome if you need to be highly expressed in males, which is kind of weird because Drosophila males have a way of dealing with a haploid X chromosome, okay? So Drosophila males should only express X-linked genes at about half the level relative to females would be expressing them because they only have 1 copy. But there's this dosage compensation complex, which I think many of you are familiar with, that assembles specifically in male Drosophila because one of the proteins is only produced in males. And this thing is proteins and RNA. It binds nearly exclusively to the X chromosome. It causes acetylation of lysine 16 and acetylH4 and promotes upregulation of expression on the X chromosome. And you can see, if you look at the polytene chromosomes I mentioned earlier, you see this dosage compensation complex staining really nicely across the X chromosome. And so we would imagine then in males, if they wanted to express genes at a particular level, okay, or they were expressing them at a particular level, the dosage compensation complex would bind, double that expression. Now what we think might be going on is there's actually some threshold, and we call it a dosage limit. And if you exceed that, or you can't exceed that basically, for some sort of mechanistic reason, maybe Leighton could come up with a mechanism, but there's evidence from other systems where there might be just sort of a maximal limit. And so you basically hit that threshold. And so if you want to be highly expressed, even if you're dosage compensated, you still can't exceed that limit. And so the X chromosome is not a good place to be. And so we think maybe there's a dosage limit, even with the dosage compensation system in Drosophila. So I said, right, there's a deficiency of genes specifically expressed in the accessory gland on the X. If we look at those that are on the X and specifically expressed in accessory gland, they're expressed at a lower level than the genes that are specific to the accessory gland and autosomal. So the remaining genes are autosomal. They're expressed higher, the autosomal ones. Now, it's possible, though, that dosage compensation might not even be occurring for these accessory gland genes. And so what we did is we looked at data available for the binding of the dosage compensation complex to the X chromosome and asked which genes are bound and which genes are not bound by the complex. And then we asked where are those genes expressed. And it turns out that the genes that are bound by the complex tend to be expressed broadly across many tissues. And the genes that are narrowly expressed in single tissues tend to not be bound. So there might be a bit of an ascertainment bias in how the chip data the chip experiments were performed for this, but it's possible that the accessory gland specific genes are dosage limited and not even dosage compensated, and that produces a further constraint on their presence on the X. And so that's a male-specific tissue, right? And so it would make sense then in a male-specific tissue where males have one copy of the X that you would not have the male-specific genes expressed in that male-specific tissue being found on the X because of the dosage limits. And so we were wondering, does this constraint affect tissues or functions that are not male-specific? And so for us, an obvious case of induction or high expression is the immune response to pathogens. And so Drosophila have an innate immune response. They don't really have an adaptive immune system. And that consists of recognizing pathogens, signaling, which includes toll and IMD, which I mentioned before, and then that signaling causing the induction of effector proteins, including antimicrobial peptides. And so if you look at the antimicrobial peptides that are induced upon infection, there's none on the X chromosome in Drosophila. So they're all autosomal. There's also none on the left arm of the second chromosome. So if we want to tell stories and make stuff up, sure, we can tell stories about the left arm of the second chromosome all day. So we said, well, this is a very small sample size, right? These are annotated antimicrobial peptides. There's not that many of them in the Drosophila genome. In flies that live in nastier places, there's a lot more. But can we try to understand this deficiency on the X with a bigger sample size? And so we said, well, what if we look to see what's induced upon infection independent of how it's annotated? So we just say, what genes get induced upon infection in Drosophila? And so we took this nice large RNA-seq data set that was published a couple years ago, and in collaboration with Rob Unclis at Kansas, a graduate student, Daniel, in my lab, analyzed these data, and based on the number of induced genes, he said, okay, I would expect, based on the size of the X chromosome, this many to be on the X, and I observe the bar graph amount. So with E. coli, there's actually no induced genes on the X chromosome. With E. facilis, there's maybe one or two. The E. coli is not significantly less than the expectation because of small sample sizes and low power. With facilis, there is a significant deficiency. And then we looked across the full set of 10 bacteria, and for 6 of those 10 bacteria, there's a significant deficiency of induced genes on the X chromosome. Actually, for all 10 of them, they're below the expectation. So you can calculate a probability that all 10 would be beneath the expectation, and that's very unlikely. So it seems like for response to pathogens, or at least bacterial pathogens, there's a deficiency of genes that respond on the X chromosome. We then said, well, is this specific to the individual pathogens? So Daniel said, okay, what if it's induced by at least 2? Okay, so a gene that's induced by at least 2 of these pathogens. There's a significant deficiency on the X for at least 2, for at least 3, at least 4, at least 5, at least 6. For 7, it's not significant because our power just gets too low. Seems like there's a dosage limit, and if you want to be highly expressed on the X chromosome, you hit that ceiling and you can only you can't get above it, and males, and that selects against genes that need to be induced in context-dependent ways. For example, in a specific cell type or tissue type, in a specific environmental context, such as infection. It selects against genes on the X. That's the copy number effect. So we use sort of this exception of the X chromosome to show that there's potentially some limit or constraint on context-dependent upregulation based on copy number within cells. What I'm going to do now is circle back around to, for the rest of my talk, to talk about a single genome shared across contexts. And for that, I'm going to talk about work we've been doing in housefly. And you might ask yourself, why would anyone work in housefly? And I will tell you why. So housefly has a really cool polymorphism in how it determines sex. So houseflies have a gene transformer, which if you're familiar with Drosophila sex determination, it's the same transformer gene. And transformer is alternatively spliced in males and females. So if it's spliced into a functional protein, it induces female development. If its splicing is into a non-functional protein with essentially the transcript has premature stop codons, you get male development. And the way that that splicing is regulated in housefly, and so far only in housefly, this regulator is only found in housefly, is this thing called MDMD, or Musca domestica male determinant. And it's a negative regulator of transformer. So it causes transformer to be spliced into a non-functional isoform. MDMD can be found on the housefly Y chromosome, because the Y chromosome is the male determining chromosome, but there's multiple different sort of Y chromosomes in housefly populations. And so MDMD can be found on the third chromosome as well, and we can find males in the wild that have multiple copies on, or multiple different male determining chromosomes. So they can be homozygous for the 3M chromosome, they can be they can have a YM and a 3M. And we've shown, and I'll get to this a little bit later, but I'll just plant it here, that both of these YM and 3M are what we call proto-Y chromosomes. So they're very recently derived Y chromosomes that carry a very new male determining gene. That's not the whole story. There's a second allele of transformer called TRA-D that is desensitized to the male determiner. And so a female that has or a fly that has the TRA-D allele can be developed into a female even if she has the male determiner, because the male determiner doesn't work, basically. So we can have, that means the TRA-D bearing chromosome is a W in a ZW system. So we have an XY system, a ZW system, all this variation. We're interested in understanding what's responsible for maintaining this variation. I'll talk a little bit about that now, but we also have a lot of ongoing research in the lab trying to understand why all of this exists. And one thing that we think is important in understanding this is looking at the natural distribution. So, and you sample houseflies. The YM chromosome is the only Y chromosome found in the north. So this is sampling from Maine, so even north of here. And then the 3M chromosome is the only Y chromosome found in the south. So if you go to Florida, you only find the Y, or the 3M. And in the middle, you find both of them. And so we think what's going on is that temperature might be an important factor in acting on these different genotypes and causing them to be found in this distribution in the wild, and such that the 3M genotype, we think, is warm adapted, confers fitness benefit in warm temperatures, and the YM is cold adapted and confers a fitness benefit in cold temperatures. So this would be a genotype by environment interaction. We refer to it specifically as a genotype by temperature interaction. And a graduate student in my lab, Kieran, has done experiments that have shown that if you put these chromosomes on common genetic backgrounds, you can actually get the predicted cold and heat-tolerant phenotypes in the flies. And then a postdoc, Pablo, has further gone on to show that you actually get a preference. So you, as a northern fly, might prefer to go somewhere colder, and I, as a southern fly, might prefer to go somewhere warmer. And all of this depends on developmental temperature. So Ys basically are acclimating, their physiology is changing, their behavior is changing contingent on their genotype and their developmental temperature. And so we would love to figure out what the genetic basis for all of this is. And I'll spoil the ending, and we haven't figured it out yet, but I'll tell you what progress we've made. So we have this genotype by environment interaction that we think is affecting these phenotypes to maintain this genetic variation. We think sex is an important factor as well, because the females can carry these Y chromosomes if they have that TRA-D allele. We haven't been able to do the sex experiments yet, so I'll only be able to tell you the genotype and environment work. So in order to look at the effect of genotype, we first needed to figure out the extent of genetic differentiation between the different genotypes. And so here we said, well, let's take a XX female and an XY male, and working with the reference genome that we have for houseflies that came from a female, that genome should not have a Y chromosome, it should just have the X chromosomes. A technician in my lab, Chris, sequenced a male genome and also sequenced a female genome side by side with the male, and we used a differential coverage approach to try to identify X chromosome sequences. And the idea with the differential coverage approach is that if the males have 1X and the females have 2X, then if you sample autosomal sequences from the two sexes, you should sample them equally in males and females. And so if you map those reads back to the reference genome, they should be present, mapped basically in equal amounts in males and females. And so if you take that male-female reference genome mapping or coverage, you take the log of 1 for equal coverage, you get 0. But the X chromosome is found in 2 copies in females, 1 copy in males. And so if you take the male-female ratio, it should be 1/2. Log 2 of 1/2 is -1. Okay? So if the X chromosome is differentiated from the Y, males should have 1X. The log 2 ratio should be -1. What we observe is that the X looks just like the autosomes. So males basically have 2 copies of the X chromosome. It just so happens that one of them carries a male determiner, and it makes it a Y. So they're very, very undifferentiated XY pair. So we said, well, can we find anything specific about the Y? It doesn't seem to be very differentiated from the X, but is there anything specific to it? And so we took an approach where you can use a male genome assembly and sequencing reads from females. And if you take those female sequencing reads and map them back to the male genome, but you map what are called k-mers, which are unique sequences, so it sort of gets around this issue of trying to map reads uniquely to the genome. So you just map the k-mers. What you should see is if you look at where the k-mers are mapping in the male genomic scaffolds, you should get a bimodal distribution if there are Y-specific sequences. And so the x-axis here is a little weird. It's the percent unmatched in the male genome assembly. So 0% unmatched is not unmatched, and so if you cancel the double negative, it's 100% matched. Okay, so 100% matched means it's autosomal or it's X chromosome because they're both found in males and females. 100% unmatched means Y chromosome because it's male specific. And so if you do this in a Drosophila genome, you get that bimodal distribution where you get X or autosomal sequences and you get Y chromosome sequences. An undergrad in my lab, Quang Lu, did this with our sequence data in housefly, and we did not see that peak at the Y chromosome. We saw the X autosome peak, but not the Y chromosome peak. So it seems like if there are Y-specific sequences in the housefly genome, they're very hard to find, and maybe there aren't. And so we said, well, can there be anything on the Y chromosome that's special, other than the male determiner? And so we turned and looked at those box plots I showed you before of the log 2 male-female coverage, and we said, well, what if we include the outliers in the box plots? And so on the left is the expectation if the X and the Y were differentiated. On the right is what we observed. I showed you that already. I just added the outliers to the plot. And just as a sanity check, we went and sequenced 2 additional strains to kind of say, are we really seeing no differentiation? And all 3 strains showed the same pattern. But the outliers, if we looked at them, we said, well, there are a bunch of these outliers that have really high log 2 male-female sequencing coverage, and they're above a 1.5x enrichment in males. So 1.5x on a log 2 scale is 0.66, I think. And that's where that box is circular like boxing it. That's 1.5x is consistent with a gene that is present on the autosomes and has a single copy parallel on the Y chromosome. Okay? And only on the Y chromosome because that's how it's enriched in males. Okay? And one of the genes that falls in that outlier is a positive control for being a duplicate, which is, it's a gene called CWC22. And the reason it's a positive control is this is the ancestral paralog of the male determining gene. And so the male determiner in housefly is a new gene. It's a new gene because it's a new duplicate of an autosomal splicing factor. And so we find this gene here in our enrichment in males because it's present in 3 copies in males. It's a very recent duplication, so the reads are mapping to all 3 copies in males. And so these other outliers might be interesting candidates for following up, which we haven't had a chance to yet, but potentially what makes the Y chromosome special is having recruited autosomal duplicates. So that's one of our new Y chromosomes. The other one is the 3rd chromosome. And what a graduate student in my lab, Jay, did is he took his sequencing data and he said, how's heterozygosity distributed across the genome in these strains that we have in the lab? Okay? And so we could quantify heterozygosity on a scale from 0 to 100. So 0 is our measure of the males have no heterozygosity, but the females have heterozygosity. And 100 is the males have heterozygosity, but the females do not. So 100, the top there is the males are the only ones that are heterozygous. And on the 3rd chromosome, we see that the males have elevated heterozygosity, which makes sense because these males in this particular experiment were heterozygous. They had the 3rd chromosome male determiner and then the regular 3rd chromosome. So these males are now having the 3rd chromosome male determiner, not the Y, that XY male determiner. Okay? So the 3M, we call it, and standard 3rd chromosome seem to have the same gene content. So we can't find any gene differences, but there's allelic differences between the 2 copies. Okay? So we have 2 different sex chromosome pairs, the XY I told you about, and now this 3M system, where the X and the Y are very, very similar, but there's slight sequence differences. And so if you think about the trajectory of sex chromosome evolution, you have a pair of autosomes. One of them acquires a male determiner, and then that becomes a proto-Y chromosome. And the other one, de facto, becomes proto-X. And then because of the male-limited inheritance of the Y chromosome, it's a good place for male beneficial, female detrimental alleles to be, because they can benefit males without coming at a cost to females. But in order for that to work, you need to have suppressed recombination between the X and the Y to prevent those alleles from moving onto the X and being inherited by females. That suppressed recombination leads to things like Muller's ratchet and hitchhiking, causing degeneration of the Y chromosome, in addition to the Y chromosome having a small affected population size. So humans and Drosophila are over there on the right-hand side, and housefly is somewhere around here. Okay, so it's a very young system, very little genetic differentiation. And so in terms of genotypic variation in the system, responsible for some interesting phenotypes we observe, there isn't much. Right? So there's not much genetic variation that could cause these phenotypic differences we observe. And so this presents us with a paradox where we see this distribution of these genotypes in the wild. We see that they have phenotypic effects in the lab, but there's not much genetic differentiation between the YM and the homologous X and the 3M and the standard 3rd chromosome. So how are they causing the phenotypic effects? And that's, I'm going to disappoint you. I'm not going to be able to tell you everything, but we have some ideas. So the approach we've taken for this is to measure the phenotype at sort of the most basal level, which is gene expression, right? That's sort of the first step of the manifestation of genotype And so we can take a YM chromosome and a 3M chromosome, put them on a common genetic background in the lab, and compare gene expression. And we published a couple papers doing this. The first one we published, we saw that we have an expectation for the percent of each chromosome that should be differentially expressed between the two genotypes based on the number of differentially expressed genes in the genome. And we see that on the third chromosome, when we compare these two genotypes, there's an excess of differentially expressed genes. Well, duh, they have differences in their third chromosome genotype, right? The part of the genome that's genetically different is also the part of the genome that's differentially expressed between the genotypes. The X chromosome looks like it might be in excess. There's actually not very many X chromosome genes. It's a really weird chromosome. It's the Drosophila dot is the X chromosome, so we just basically have no power to do anything on the X in this system. So the question we next asked was, we see expression differences between these two genotypes. Are they unique to putting a new Y chromosome essentially on the genetic background? Or can you do it by putting any darn chromosome you grab? And so we said, well, let's just take another third chromosome that doesn't have a male determiner, put it on the same genetic background, make the flies heterozygous. What happens? And so Jay did this experiment, and I just published it last year. He said, well, let's compare YM and 3M and count the number of genes that are differentially expressed. And he did two different comparisons, and it's pretty similar in the two comparisons. You compare 3M versus 3M, so two different 3M strains, it's fewer. So 3M genotypes seem to be more similar to each other than they are to the YM genotype. But if you take that YM genotype and just throw a new third chromosome on, you can basically get the same amount of differential expression as if you put the male determining third chromosome. So there doesn't seem to be very much special about these male determining third chromosomes in terms of how they affect gene expression relative to any old third chromosome we happen to have in the lab. And so, once again, we're presented with this paradox. We have very little genetic variation, very little phenotypic effect in terms of gene expression, yet a phenotypic effect observed both in terms of the natural distribution and in terms of the lab phenotypic assays. So we said, well, can we do this control for background even better? And so what we did is we said, well, let's manipulate the sex determination pathway and use double-stranded RNA to knock down transformer and take a genotypic female, convert her into a phenotypic male, and ask, if you're phenotypically male and you have the Y, or a Y, which is the third chromosome here, or you don't, how different are you in terms of gene expression? So the design of the experiment is a little more complicated than that because we had a sham control as well. So we injected with GFP targeting double-stranded RNA targeting GFP. There's no GFP, this shouldn't do anything. And then we targeted the transformer gene that causes the sex reversal, and then Jay measured gene expression in all four of these phenotype by treatment combinations using RNA-seq. And when he ran a PCA on his gene expression results, the colors indicate treatment, the shapes indicate genotype. The thing that clusters is phenotype. So phenotypic females all cluster together. Males cluster together based on phenotype, not based on genotype. So once again, that 3M chromosome, you can take it out of a male, he looks like a normal male. And we tested these males, they're fertile. Like, they're perfectly happy without any Y chromosome. And Jay could count the number of differentially expressed genes. So if you compare a normal male to a female, in the tissue sample here, you get that many differentially expressed genes. You take that sex-reversed male that's genotypically identical to the female, but at the gene expression level has just as many differentially expressed genes as a normal male compared to a female. When you compare two males, even if they're genotypically different, they're quite similar to each other on the same amount, basically the same level of similarity to each other as if you take two genotypic males and compare them to each other. So the one thing that could be interesting is to see what specific, so wholesale there's not a big change, right? but there might be specific individual genes that are affected by this genotypic swap out. And so this is all the genes in the genome sort of colored by whether they have sex-biased expression in males or females, so up in males or up in females. You can see some individual rows there that are different. And so Jay pulled those rows out and said, okay, here's one example of one of those rows where the gene is higher in sex-reversed males than females and lower in genotypic males than females. So basically we call them discordant sex-biased genes. They go in opposite directions depending on male genotype. And so this is all of them for this particular tissue sample, and they're enriched on the third chromosome. So it's possible that there's individual genes that are important for conferring these phenotypic effects, but there is not a wholesale reshaping of the gene expression profile in these organisms based on these Y chromosome genotypes. So to dig in this a little bit farther, Jay's been trying to see which of the chromosomes these genes are being differentially expressed from. And so what we have is a third chromosome male who should have a bunch of genes with a bunch of cis-regulatory elements that are regulating their expression. And if there's a chromosome-wide change in expression, we would expect to see a lot of cis-regulatory divergence in third chromosome genes. So the red is indicating they have new cis-regulatory sequences that are affecting their expression. In contrast, if there's individual alleles that are differentially expressed and potentially have large effects, maybe they're modifying expression or function of other genes on that chromosome and throughout the genome, then you should only detect individual genes with cis-regulatory changes in their expression. And so to look for cis-regulatory changes in expression, we tested for allele-specific expression between the third chromosome and the 3M male-determinant chromosome. Okay? So Jay took his data from the genotypic males and sex-reversed males, and basically operating under this assumption that if there's a chromosome-wide cis-regulatory effect, so a lot of cis-regulatory changes on the 3M chromosome, then you expect to see an elevation of allele-specific expression in those 3M males relative to the sex-reversed males that should be homozygous. Okay? And so these two plots here is showing genes with allele-specific expression in the 3M males, so differential expression between the 3M and standard third chromosome, but not differential expression between the two copies of the third chromosome in the sex-reversed males. And on gray is genes on the third chromosome, and as sort of a negative control in the analysis, we looked at genes on the rest of the chromosomes. Those shouldn't have any real biological signal. And so anything in excess of the white is potentially interesting. And then he compared it to the opposite allele-specific expression pattern. So not allele-specific in the heterozygotes, but allele-specific in the homozygotes, which that shouldn't be interesting. And it's nice that that's less than the white bar. And there's a significant excess of allele-specific expression on the 3M chromosome, but it's very moderate, right? And there's not a lot of genes, which is consistent with not a lot of genes being differentially expressed, and consistent with a lot of genes not having discordant sex-biased expression. And in fact, Jay found one gene which had discordant sex-biased expression and discordant allele-specific expression. So it's upregulated in the 3M males, and it's differentially expressed between the 3M and standard third chromosome. It's called HEAT2 in Drosophila, or HEAT-R2. I don't know, we need one of those pronunciation guides for gene names. So there's 3 diagnostic variants in this gene that allow us to test for differential expression between the 2 chromosomes. And if you look in the 3M males, you can quantify the expression at each of those diagnostic variants for the standard third chromosome allele. And then you can, so that's the green triangles at the bottom. Those same 3M males have a second allele, this is the green squares. That's the expression from the other allele. And if you add those 2 up, the circles are the whole gene expression from both alleles in the 3M males. And then the purple is if you're just a sex reverse male, so you don't have the 3M chromosome. And so this gene is upregulated in 3M males because the 3M allele is upregulated in its expression. So why would the 3M allele be upregulated in expression? So what does HEATR2 do? It's involved in accidental diamond complex assembly, which is important for sperm motility in flies. It's also important for ciliated mechanosensory neurons. I know nothing about this. There was just a paper that said that. And so our hypothesis is that there might be an intersexual conflict over the expression of this gene. And so potentially overexpressing the gene could, in non-spermatogenous cells, could be deleterious. And so there's selection in females to prevent it from getting expressed too high. But if it's Y-linked, you can express it higher. That's just a hypothesis. Very hand-wavy one at that. So there's some effect of the genotype on expression. But what I haven't said as much about is at the molecular level, how is that environment dependent? So I'll kind of close with that. So we have this environmental, or this latitudinal gradient of these different genotypes. We think that temperature is an important factor in maintaining this gradient. So can we detect the effect of temperature on these genotypes? And so what we did is we took our two different genotypes, we raised them at two different temperatures. This is kind of the thermal limit that we can raise these guys at, so we kind of push them to their extreme. And if we looked at the sex determination pathway, there's no meaningful differential expression in the pathway. So it's not like some other systems where you have a temperature-dependent sex determination pathway, and that's what's under selection. We think that what's going on is that there's alleles on the proto-Y chromosomes that are affecting phenotypes in a temperature-dependent way, independent of the pathway. So this was an experiment that was done by another graduate student in the lab, Kieran, I mentioned earlier. So this is his principal component analysis of his experiment, where he looked at the genotypes at the different temperatures. And if you squint, you can kind of see that if you take your northern Y chromosome and raise it at the warm temperature, and your southern Y and raise it at the cold temperature, they sort of separate along principal component 1. They're wrong. You take the northern Y chromosome and raise it at the cold temperature, they separate along principal component 2. The southern Y chromosome at the warm temperature as well, and then the opposite. So the unpreferred, you take the northern one to heat, the southern one to cold, they separate along PC1. You see some of this pattern in clustering. So if you look at the clustering of the gene expression profiles, they cluster by genotype. There's a little bit of clustering by temperature after genotype, but it's kind of messy. So there's not a very strong signal here of genotype by temperature interactions. If you compare genotypes at each of the temperatures, the chromosome where they're different in their genotype is the chromosome that's the most differentially expressed, like I showed you before. Not all that interesting. What surprised me a little bit is when you compare a single genotype at two different temperatures, you don't see any enrichment on specific chromosomes, which is consistent, once again, with these chromosomes not having a large-scale effect on gene expression on that particular chromosome. And so this is consistent with what we were finding before, is that there's not a large-scale change in cis-regulatory sequences driving gene expression differences between these genotypes. And so Kieran kind of did something similar to what Jay had done and looked at allele-specific expression between the two different chromosomes in the 3M males. And so what I'm showing you here for 3 genes in this graph is the difference in expression between the 2 chromosomes at each of the temperatures. And so that blue dot I've got pointed out there is a situation where the 3M allele is upregulated at 18 degrees, and then here the 3M allele is more downregulated at 18 degrees. And so Kieran identified genes where there was differential expression and allele-specific expression, and asked, is the differential expression that depends on temperature consistent with the allele-specific expression that depends on genotype or temperature? And we would expect that if cis-regulatory alleles were responsible for the gene expression differences we should see allele-specific expression that's consistent with the temperature differential expression based on genotype. And so the top row is the 4, for 4 genes have that consistent pattern. And the bottom row is 4 for 4 genes do not have the right pattern. Okay? So 4 genes have the right pattern of allele-specific expression and differential expression, and 4 genes do not. So Kieran was like, okay, that's the 3rd chromosome. I don't expect any relationship in the rest of the genome. Turns out the rest of the genome has a much nicer signal than the 3rd chromosome. And so once again, we think that wholesale cisregulatory modification on the 3rd chromosome has not happened in the system yet. Right? There's only a few large effect changes. And so all of these results so far are consistent with this hypothesis, where there's individual alleles with large effects, potentially, on the 3rd chromosome that are affecting these phenotypes in a context-dependent manner. And there's not a wholesale change in the regulation of this chromosome that's driving this genotype by environment interaction. So there's a minimal genetic differentiation between both of our sex chromosome pairs. The phenotypic effects seem to be environment dependent based on organismal experiments we've done, and our gene expression work seems to suggest that those organismal effects are driven by a small number of genes that have potentially large phenotypic effects, and I would love to tell you what those genes are. We have some candidates, but at this point we don't really have a solid proof of any of them. We're working in housefly, so we might basically have just reached the limit where we can go in this system. And so what I'm interested in going forward is moving some of this work into Drosophila and identifying patterns like this and being able to actually functionally dissect these interactions. And what we haven't done yet in housefly, which we are going to be able to do, is look at the effect of sex as well. So I told you everything was done in males so far. So we can put these Y chromosomes into females because of the weird sex determination system and ask if these G by E effects are sex dependent. Okay, so in various levels of detail and with various amounts of data, I talked about 3 constraints on context-dependent expression. Talk a little about how small sex chromosome differences can give rise to phenotypic differences between the sexes, even though most of the genome is the same. I presented some evidence that there's an X chromosome dosage limit that affects both male-specific and traits shared by both sexes, such that if you need to be an induced gene in a specific context, it's probably X chromosome is not a good place to be. And then in housefly, how Y chromosome polymorphism can affect gene expression and potentially has an ecological relevance there, but it's very minimal effect. So going forward, looking at developing some functional genomics approaches that will allow us to do comparative studies. So we can do comparative genomics, we can do functional genomics. It's really hard to do comparative functional genomics, as some of us have tried to do, and trying to develop statistical methods for doing that. Also looking at this shared signaling pathways hypothesis I presented, and then also this looking more at the genetic architecture of phenotypic plasticity and how it relates to context-dependent expression. So once again, these are the people that have collaborated on this work. Thank you very much for your attention.
For most of the work where you weren't growing the houseflies at 19 or 18 or 29, were you growing the YM and the 3M at the same temperature?
Yeah.
And were the flies then, if it was, let's say, the middle temperature between their preferred temperatures, were you stressing them both, one cold stressed and one hot stressed?
That was before we realized how important temperature was. And so we were growing them both at the same temperature in my colleague's incubator that he had. They were lab adapted for that particular temperature, but probably not fully adapted, as we found in these dark turbulent temperatures. But yeah, they were all at a warm temperature because that's how they raised them in his lab. But now we're realizing this effect of temperature, and maybe humidity is important too, which is something we're.
Well, that was the follow-up question.
Yeah.
That's not the only thing that's different between Maine and Florida.
Oh yeah. No,
I would think, for example, pathogens could be very different, which would be perhaps a temperature dependent, but a secondary effect that really is the inducer that you should be using?
Pathogens.
Just as an example.
Yeah, no, I think that's a good point. And the reason we chose temperature is as a fly lab, so temperature is different. And as a fly lab, we have multiple incubators, and we can set them to temperatures and manipulate the system that way. And so it was very much a tractability thing. We were getting organismal response at those temperatures. We were getting we were interested in the gene expression effects. But yeah, I mean, the problem we have is sort of the opposite of a lot of other systems, where we have a known genetic difference and no known phenotype, whereas a lot of times when there's some sort of ecological relevance, there's a known phenotypic difference, and people are trying to find the genotypic cause. And so phenotypic space is very broad. Yeah, but I agree totally. Like there could be a whole bunch of other things going on. Yeah, sorry.
Yeah. So you mentioned it's not the X chromosome is not a good place for an inducible gene to be in. So have you or anybody ever tried to move inducible genes to the X and see misregulation, either sex specific or not, and some sort of fitness cause?
That's one experiment that my colleague Rob, who's working on this together, we'd like to do. So there's particular Drosophila antimicrobial peptides that there's very clean phenotypes when you knock them out with particular pathogens. And so we want to ask, if you try to rescue that, does that rescue depend, the effect of that rescue depend on where you put it? Yeah.
So you said there was a dearth of infection-induced genes on the Drosophila X. Right?
Yeah.
If I understood what was going on in the housefly, I would guess you probably wouldn't expect to see that in the housefly.
Yeah.
On this proto-X. Is that true? Have you tested that?
We have not tested it. We wouldn't expect it, and we could test it. We should test it. And I was going to say one other thing there. It becomes so I'll caveat the Drosophila work that when your sample sizes get small enough, there's a bit of sort of evolutionary contingency, and so we're trying to. That's why these functional assays are important to validate. But yeah, I think in housefly we should look at that. We should expect a random distribution.
Your Kamer approach, you talked a lot about genes and gene expression. Did you actually allow multiple high copy number Kamers? So one of the things that's one of the theories about incipient chromosome evolution, particularly with sex chromosomes, is that emerging sex-determining systems actually will be surrounded by heterochromatin blocks and highly repeated sequences. And I'm wondering, is there anything else other than gene expression that may give you some insight into the evolution of these proto-sex chromosomes?
Yeah. So the housefly male determiner is a hell of a locus. So it's a tandem array of a particular gene. We don't know if one copy is functional or all of them. It's tandemly arrayed wherever it's found too. It's surrounded by presumably heterochromatic sequence because it's highly repetitive. So it's surrounded by repetitive sequence. Makes it hard to CRISPR out. Makes it hard to genotype the animals for which genotype they have. We've looked at the expression of it compared between the genotypes, and there doesn't seem to be a meaningful expression difference of that gene. Protein sequence-wise, because it's a tandem array, it's hard to know which paralog to look at, but there's not a lot of differentiation. I don't think there might not be any differentiation in terms of protein sequence between the copies on the different chromosomes. So it seems to be, for everything we've done, it seems to be a functionally equivalent regardless of where it is.
What about the repeats?
So if you do your PCR right, and you put primers in unique sequences, you can kind of get like essentially different AFLP patterns from the different genotypes, but you move that to another lab and it's not repeatable. So yeah, we've got some PacBio sequencing now and Nanopore, so we want to sequence across this whole thing, and that'd be nice to have a better sequence. We also that X chromosome, the ancestral X chromosome of flies, is the Drosophila dot, which is itself this sort of heterochromatic zone that only has 80 genes on it, that has all sorts of weird regulation. There is a dosage compensation system in flies outside of Drosophila that could potentially be interesting. Anyway, sorry, I'm like rambling
Can you
now,
look at
but
chromosomes for these guys though? I mean, could you just do like a
can I?
old school, anybody, like comparing genome hybridization?
People
so you
can.
might
So,
see a
so
copy
actually, I mean,
number?
if you go back and read Nettie Stevens' turn of the century papers, housefly is in there.
Yes.
So like this is that the reason it's called the X chromosome is because it's stained as constitutive heterochromatin in flies, right? So like this is not spermatogen or not MSCI, right? This is like constitutive heterochromatization. So yeah, if you can. Me personally, that's a skill set I would like to have.
When you were talking about your allele-specific gene expression on chromosome 3, one of the first things that came to mind to me was cystic regulatory elements. But you mentioned this might not be likely based off of the...
Large scale. So there might be individual genes that have divergence.
That's what I was going to ask, if you've gone into some of those
Yeah.
genes and looked at their promoters or enhancers nearby to see if there's...
Yeah.
also sensitivity
So the problem we run into in non-model organism genomics is like the annotations don't exist. And so really if you look at like our annotation files, it's just protein coding sequences and like TRNs, shit like that. Jay has gone and looked at where predicted transcription factor binding sites would be upstream of that one gene Peter 2. And then he has some nanopore sequence data which he used for his allele-specific expression to do phasing, but then he also looked at the nanopore data upstream of the gene to ask where are there SNPs upstream of the gene that could regulate expression, and there's like 3 SNPs and they're not within predicted transcription factor binding sites. So we've tried, but it's like it's not. You sort of reach a tractability limit in these things. That's one reason why Drosophila